Today, we are going to discuss the difference between dextrose, dextrin, and dextran. What is the main difference between these three molecules that we will look? So first about the dextrose. This dextrose word that is also commonly utilized in clinical practice. So dextrose is a common name used for the D-glucose. So D-glucose, that is the main glucose molecule present in a blood and also the main source of the energy. So that simple D-glucose is called as dextrose. Here a structure of that D-glucose, that is simply C6H12O6, but here the arrangement that is making a D-glucose. Similar type of structure, that is L-glucose, but it is not present in our blood. Now, so simple you have to remember dextrose, that is D-glucose. Next, that is dextrin, that is simply oligomer of glucose which is produced as an intermediate molecule that is produced by the enzymatic hydrolysis of the starch. So starch hydrolysis, that is making a different molecule of the dextrin. So this dextrin is also subcategorized in a various molecule, that is amylodextrin, erythrodextrin, and acrodextrin. Why that different name is given, that we will look. So starch, that is basically react with the iodine and giving a blue color. Further hydrolysis of the starch, that is produce amylodextrin. This name is given because amylodextrin react with the iodine and giving a violet color. Further amylodextrin hydrolyze and giving a red color with the iodine. So name is given erythrodextrin. Simple you have to remember, erythro means red color. Erythrocyte means red blood cell, that is erythro means red color. Further hydrolysis of erythrodextrin, that will produce no color with the iodine, and so name is given acrodextrin. So that all dextrin, these three main dextrin are called the dextrin molecule, are the enzymatic hydrolysis of the starch. Here a structure of this dextrin is shown. So that basically it is made up of this straight chain that is made up with the alpha 1,4 linkage, and branch in this straight chain that is made up with the help of alpha 1,6 linkage. So straight chain is made up with the help of 1,4 linkage, and branching that is occurring with the help of alpha 1,6 linkage of the glucose molecule. So these two, the difference between dextrose and dextrin. One more molecule, that is dextran. It is a polymer. Main chain consists of the alpha 1,6 glycosidic linkage between glucose monomer, while branches that is developed at the alpha 1,3 and 1,4 linkage. So the characteristic branching, so particular branching, that is 1,3 and 1,4 linkage, that is distinguish a dextran to the dextrin, which is a straight chain glucose polymer tethered by the 1,4 and 1,6 linkage. So you have to remember dextran, it is straight chain made up with the help of alpha 1,6 linkage of the glucose monomer, while branching at the 1,3 and 1,6 linkage. That is making difference between dextrin and dextran. Further, this dextran, that is a high molecular weight. It is consist of the 1 to 4 millions, and that is making a bigger size molecule, and this characteristic is utilized for the treatment purpose. Mostly this bigger size molecule that cannot escape out of the vascular compartment, so it is given in a hypovolemic sac as a plasma volume expander. So this dextran is also commonly utilized in clinical practice as a plasma volume expander, mostly given in a hypovolemic sac. Here a structure of this particular dextran. Here multiple unit of the alpha 1,6 unit, that is making a straight chain, while branching develop at the 1,3 linkage. So that is the main difference between dextrose, dextrin, and dextran. So you have to remember which molecule is this. This one, that is simple a D-glucose, or called a dextrose. This one, that is 1,4 linkage straight chain and branching at the 1,6 linkage, so it is a molecule of the dextrin. Further, there is a chain with the help of 1,6 linkage and branching at the 1,3 linkage, so it is called dextran. Now which one is this molecule? That is C6H12O6, that is instead of this dextrose, it is a L-glucose. So dextrose, that is D-glucose, similar type of structure, that is L-glucose, but it is not present in our blood, or it is not the main source of the energy. So that's all about the difference between dextrose, dextrin, and dextran.